Okay, so now we are going to tackle your carbohydrates, which is now your module 5. So for this, your carbohydrates, as we all know, is the source or the main source of our energy from our prelim. It is produced in bulk, usually by organisms, especially our plants. So we people or we humans consume a lot of your carbohydrates in order for us to produce energy, which is our ATP. Your ATP is derived from the catabolism process of your sugar, which is known as your glycolysis. So after your glycolysis, it will undergo your Krebs cycle, and then it will undergo your ETC, electron transport chain, and all of that. So we'll be tackling that. And so in here, it is known that this is the first product formed in our photosynthesis, and it is usually a structural component also of many organisms, especially your plants, which is your cellulose. Okay, so let's move on. So a normal carbohydrate is actually soluble and in crystalline form whenever they have lower molecular weight, and for higher molecular weight they are usually less water soluble and amorphous. So when we talk about amorphous, they are kind of powder like, okay, and tasteless. So examples of your low molecular would actually be your normal table sugar or our sucrose, our glucose, and for the high molecular weights, these are our starches. Okay, so these are your potato starches or mga different na mga harina. So as you can see, it is amorphous, flour type, it is in powder type, and when you taste it, it doesn't taste sweet. And when you dissolve them into water, if it is too saturated, it becomes less soluble. So compared sa aton nga sugar nga normal or our table sugar. So next, we have this different classifications of your sugar. We have your monosaccharide, disaccharide, oligosaccharide, and polysaccharide. And if you have to study this one, of course, by the word mono, it means that this is composed only of one sugar form. Okay, so simplest form of sugar. And in your disaccharide, this is the combination again of your two monosaccharides. For oligosaccharides, these are combinations of 12 monosaccharides, not more than that. Because when we talk about more than 12, that would be now your polysaccharides. So, examples of your monosaccharides, as we know from our prelim, would be your glucose, galactose, and your fructose. So, in here, these are just the definitions of your monosaccharide. So this is the general formula of your monosaccharide, as you can see it. So it's your CnH2nOn. It means that the N here is the number of your carbon. So if there is 6, for example, carbon present in your monosaccharide, it means that you have to multiply this into 2 in order to get the number of your hydrogen, and you will get again the 6 for your oxygen. So basically your glucose is your C6H12O6. Okay, so it is your simplest sugar, and your different sugars could be uh, classified again into aldehyde or ketone depending on the functional group that you can find, okay, on its structure. So we will look at the different functional group later, and it is characterized again by the number of carbons, and the least number of carbon that you can find in a sugar is actually 3. Okay, so it is linked by your glycosidic bond, and halin pa sang prelim, sulit-sulit, your glycosidic bond is formed by a hydrolysis process. Okay, so it means that your I mean dehydration process. Okay, so into which you are going to draw out again the water, but is hydrolyzed by your acids, not by bases. So in here we have the different kinds of your sugar according to their functional group. So you are called an aldose if you do have the aldehyde group. What is now the aldehyde function group? So let's go back to your organic chem. So an aldehyde is someone that has your CHO on its structure, the CHO. CHO, remember? So it is your C double bonded to O and the presence of your letter H. In your ketose, this is actually the presence of your carbonyl group or the ketone itself. It is your carbon bonded to your O. So we have this different kind of sugars according to the number of carbon under your aldose. So if you have 3 carbons, that would be called trioses, tetros, 4, 5 if pentoses, and 6 if it is an ahexose. So in here, if you are a ketone and you have 3 carbon, you are called, or generally called, your triolose. Tetrulose if 4 carbon, pentulose, hexulose, octulose, so on and so forth. But the most important that you should really take note of would be your here, the ribose here, the xylose, and all of your hexoses, and in here the triolose and your fructose in your ketose. These are your important sugars that you should really take note of. So let's move on. So these are the functions, or these are, I mean, the structures of your aldoses. So aldoses here. Okay. So since these are your aldoses, all of them contain your CHO. But the most important of them would be your hexoses. Okay, because your hexoses would be one of your uh, one of our components into making the energy. It is one of our precursors sa aton nga mga reactions. Okay? So, no need to really memorize, but usually it is asked in your comprehensive exams, like, what is the simplest aldose sugar? What is the simplest? So, of course, the simplest would be the sugar with the least amount of your carbon, which is your glyceraldehyde. And with your ketose, so it's also asked, what is the simplest ketone sugar? That would be your dihydroxyacetone. So from dihydroxyacetone, it would lead up to many carbon again, and then you will have your fructose. So usually the most rampant or the most common form of your sugar would be for your D-stereoisomer. The right fructose, the D-fructose, your D-glucose, D-galactose, or they are more abundant rather than the L ones. So these are your stereoisomers or your enantiomers also. So here, we also have different structures of monosaccharides. So we have actually 3. We have the Fischer projection, we have your Haworth projection, and we have the third one, which is your chair conformation here. Okay, chair conformation. So Miss, nga galanlain ng structures. They actually differ in structure during chemical reactions. So if you look at your sugar simply, if you just want to isolate a certain sugar, for example, pinaka-common na lang would be your glucose or your galactose. When you just put it clearly under a microscope, so it would look like this. It would be in a linear form, so it does not react to any kind of sugars. So it will be an open chain structure, and 2D, two-dimensional. You can see it in a two-dimensional view, but the story changes if a certain sugar is exposed to water, for example, or it will be made to react to another sugars. Okay, so now that's the time that you're going to use the conversion of this linear structure from your Fischer going to your Haworth structure. Okay, so your Haworth structure looks like this. Okay, it exists into a pyrene ring or in a furan ring. So when we talk about a pyrene ring, it is a ring consisting of your 6 carbon sugar. Your furan ring is actually represented usually by a 5 ring sugar or 5 carbon ring, but usually we can use it to some of our sugars nga may 6 carbon man. So we will have the examples later. And that's the significance actually kung aaga chain structure ta, and at the same time we are using the ring-like structure. So it's all about the contact of your sugar with water or other chemicals. So they coil up and is represented by a ring. But if it is inert or it is just studied as is, so we are drawing it into an open chain structure. Okay, gets lang? Okay, we'll proceed. So with this, before converting any kind of structure of our galactose, our monosaccharides into Haworth projection, we have first to understand why is it that a certain monosaccharide here, such as your galactose, could be converted into this one. So, paano ni siya i-convert sa amo ni nga bagay? So, we'll have that in a separate video. Okay, so as of now, we have actually this theory into which we have the formation of your hemiacetal or your hemiketal. So magsiling ka hemi, it is actually half. So with your basic structure here, okay, so let's take for example your glucose here. So please memorize almost all of the structure of your monosaccharides, your glucose, galactose, and your fructose, the linear one. So from your linear structure here, you could recognize your glucose because it actually has your 1, 2, 3, 4, 4 hydroxyl groups on the right side. Okay, so it is represented by the CHO. It means this is an aldehyde. So if this is what the glucose looks like in a linear structure, it would be converted into Haworth projection if it comes in contact with your water. So now this is where this formation of hemiacetal will come in place. So as you can see, in here, your D-glucose, the the fifth carbon in here has a hydroxyl present to it. Okay, the hydroxyl carbon present to it. This oxygen will react with your carbon here in your carbon number 1 if it comes in contact with water or other chemicals. And then what happens? Usually, so I don't think I have the usually your carbon is actually double bonded to your O and then it is singly bonded to your H. So what happens now if this O reacts with the carbon number 1 here, it will break down the double chain or the double bonding of your carbon to your oxygen. So this is the time that your O here will attract hydrogen molecules from the water. By react by the time na nag-react na si oxygen to your carbon here, the H in here would also be released. So when we look at it in a fashion here, sa ato niya Haworth projection, as you can see, Miss, this is the carbon number 1, this is the carbon number 2, 3, 4, 5, and 6. Why is it nga dapat ang number 6 diri siya natupa? So in here, the O here represents the connection of your carbon number 1 and carbon number 5. And as you can see, there is non-existent or there is no existence of your OH in here. Okay? Sa first carbon mo. But when you look at in your Haworth projection, may OH na. Okay? And then in your supposedly carbon number 6, oxygen na lang di ang nabilin. So we will discuss further on how we convert this one. But the theory behind nga nag-amoni ang tsura sang imo nga Haworth from your fissure would be the formation of your hemiacetals and your hemiketals. So your hemiketal will be found in the conversion of your fructose from its linear structure going to its Haworth nga production. So usually when we produce a hemiacetal, it's just your aldehyde. Your aldehyde is the group that you can find from the carbon number 1. So it is a carbon attached to the double bond O and then attached to a hydrogen, and then the alcohol that is in the carbon number 5 in your glucose here, the OH will react to the letter C or to the carbon. And then as you can see here, the H here would be released and the oxygen will react to the carbon directly. It will break the double bond of your oxygen here to your carbon, and then the oxygen here will now attract the hydrogen from your water. Okay, so that is the same process with your fructose. So next, your chair conformation is easily converted from your Haworth if you already made the Haworth. It's just that lain lang ang pagdrawing sa iyang shape. Okay, so we have this one. If this is now your Haworth, okay, if this is now your Haworth, you will now have the chair form of your glyco or your glucopyranose. So pyranose siya in the sense that we are using a pyran ring. Okay, so this is still glucose. Naglain lang ang ngalan because of the pyran ring. So in here, as you can see, we still have the carbon number 1 here, carbon number 2, 3, 4, 5, 6. So ginlain mo lang ang pag-present sa iya. And this is how you do it. So it's either sa Fisher to Haworth mo, you use a furan ring or a pyran ring. So it's up to you to read the different characteristics of your furan and your pyran. So in here, this is the main structure of your glucose, the one that we are talking about. Ato mga aldehyde kag ginabala. Again, this is the functional group. This is your carbon number 1. This is your fifth carbon. In the formation of your hemiacetal, the OH here in your carbon number 5 would react to your carbon in your carbon number 1. It will break the bond from your double bonding. And what happens is the O here, which is now singly bonded to your carbon, would attract hydrogen from your water. So that's the time nga may OH ka na di. And then this time, the OH here will, the H here will dissociate. And the only one left would be your letter O. So this is the time that when you convert it into your Haworth, your O here will connect your carbon number 1 and 5. So when you look at it, so from linear here going to your Haworth projection, this will now represent your carbon number 1, 2, 3, 4, and 5. And this is the sixth carbon. So we did not write it here because it will not represent how the glucose will chain up, okay, or how the glucose will close its ring. So your carbon number 6 or your carbon number 6 here would be written in here in order for us to represent nga na-connect mismo ang or na-close na mismo ang rim via your carbon number 1 and carbon number 5 due to the formation of your hemiacetal. Okay? So is that clear? Okay. So for your Fisher projections of your monosaccharides, so these are your glucose, galactose, and your fructose. Please memorize the structure. So for your glucose or your D-glucose specifically, it has OH, 1, 2, 3, 4 OH on its right side. The galactose has 1, 2, 3 OH only on the right side. And in your fructose, it is different to that of your glucose and your galactose because take note, fructose is a 6 carbon sugar, but what differs is actually the second area into which this is a carbonyl. Okay? So this is therefore a ketone. Okay? So si fructose tiyak ketone na siya. So in here, we have your L-glucose and your D-glucose. So these are what we call your enantiomers. It means that they are mirror images. So they are non-superimposable. Magsiling ka ganit, non-superimposable. Kung itapal ko si D-glucose sa L-glucose, they are not equivalent. But when you put a mirror in between them, they are equivalent. Okay? So magsulat ka sa D-glucose mo as is. So if you want to write an L-glucose, what happens is that sulion mo lang. Ang apat to ka OH nga ara sa right, you put it on the other side. Okay? So next, let's discuss the terms that we are using whenever we talk about sugar para hindi ta ma-confuse. So again, here's the enantiomers. So ang bag-o ta lang again storyahan, the L-glucose and D-glucose would be what we call your enantiomers. So they are stereoisomers. When we talk about stereoisomers, they are compounds having the same molecular formula. It means like C6H12O6, your glucose could have 6 carbon, it could have 12 hydrogens, it could have 6 oxygens, but they could differ in your structural form. So they differ in the sense that the other would be dextro and the other would be levo. Okay? So they will now be called enantiomers. These two are enantiomers. And this one also, your glyceraldehyde, the simplest aldose sugar. The OH here in your D-glyceraldehyde would be on the right side, and in L-glyceraldehyde would be on the left side. Therefore, they are enantiomers. But we have other terms such as your epimers. Your epimer is still a stereoisomer term, but specifically, ang epimer taya is that they only differ at one chiral carbon. It means that, for example, this is your glucose in between. Glucose ni siya ang ara sa aton tunga. So it means that this structure in here, sa kilid ta, is an epimer of your glucose. Because this structure, kung makitaan mong ila structure, sa carbon number 2 lang sila may kinalain. So I think this is galactose. Okay? Next. In here, we have your mannose. So, your mannose and glucose are epimers at carbon number 1, 2, 2, 3, 4. So they differ only at carbon number 4. Okay, so enantiomers is your mirror-like image but non-superimposable. When we talk about epimers, they have the same molecular formula but they differ in one chiral carbon. Okay, there's a difference on the positioning of functional group sa ilang one carbon. Okay, next we have your anomers. Your anomers, it's still under your stereoisomeric theory, but we are talking about anomers when we talk about chains. I mean rings. Okay? So when we talk about anomers, we are talking about the position of your OH in your primary carbon. So usually when you write your Haworth projection, here, 1, 2, 3, 4, 5, 6, usually ang mukha ni mag-convert. If this is your alpha carbon, or I mean, if this is your primary carbon or your first carbon, the position of your OH would determine if a certain compound is alpha or beta. Okay? So if the OH goes down, it means that this is alpha. Okay? As you can see, alpha D-glucopyranose. If the OH is on the upward position, this will now be your beta. So, anong ginabasihan tani? We are actually using the term trans, T-R-A-N-S, and in here we are using the word cis. So, your OH in respective to your carbon number 6, as you can see here, they are of opposite position with each other. So, si carbon number 6 ko ang OH siya, ara sa babaw. Si carbon number 1 ko yung OH ko ara sa dalom. So they are in opposite of each other, thus the word trans, opposite. So this is alpha position of your carbons, or your OH I mean. In your beta, as you can see, in respective to your carbon number 1 and respective to your carbon number 6, their OH are both facing upwards. It means that they are cis together. Cis in position together. Magsiling ka CIS, cis, you are in the same position. Cis ka mo, di ba? Migahay ka mo. So, parehas ka mo, position, parehas ka mo, goals, di ba? So, this will now become your beta. So, kung di nakabutang si OH, kung dalom or babaw, amo na siyang ma-determine if a certain glucose or if a certain compound or certain sugar would be alpha or beta in a ring-like structure. So, that's how we differentiate all of them. So, in order for you not to be confused, in here we have examples. So when we talk about again enantiomers, they are your mirror-like images. They are again your mirror-like images into which your D-glucose and L-glucose are enantiomers of each other. Okay, enantiomers of each other. So it means that they do have the same molecular formula, but they don't have the same positioning of your OH and your H. Okay? When we talk about stereomers, kasampot ba? So, they are non-mirror image and non-identical stereoisomers. So, they do have, for example, the D-glucose to that of your L-idose. They do have the same molecular formula, but they are not equivalent. They are not mirror images of each other. And they differ also to a certain chiral carbon. Okay? So, that's it. We'll proceed. So, in here, we just have the important monosaccharides. You can read on this. So, this will just be examples. So, we have your pentose sugars, the 5 carbon sugars. We have your arabinose. So, your arabinose is actually known as your gum arabic or acacia. So, usually, your arabinose is used to form suspensions. Okay? So, kung maghimo ka mga suspension, or if you want to make, tawag sinigani, dosage forms of your drug in suspension form, you can use your arabinose to suspend your materials in the solution. Number 2 is your ribose. We do already know about your ribose from your DNA, into which your ribose is your basic sugar that is used in the production of your nucleic acid, your nucleotides, and your nucleosides. So, deoxyribose, amo man ni gyapon, specifically, we can find this in your DNA. And next, for your hexose sugar, of course, it's important to have your glucose, and number 2, your fructose. So your fructose is your fruit sugar. It means that it can be derived from your fruits specifically. So what else? Galactose. Galactose is coined as your brain sugar since this is the form of sugar that enters our brain. So amo ni siyang aton nga gina-process sa aton nga brain. So your galactose will be further broken down into simpler pagid nga mga monomers kay para ma-absorb sa aton brain. Number 4, your mannose is from your mannitol. Your mannitol is usually used in emergency situations in hospital, such as you have increased intracranial pressures. So, kung taas-taas ang pressure sa imo ulo, usually you're injected with your mannitol. So, your mannitol, when you oxidize it, it will produce your mannose. So, next, we have your disaccharides. So, for your disaccharides, we have your sucrose, our table sugar. So, it is just a combination of your glucose plus your fructose. And we are using the Hayward projection into locating the links of this one. So, if you have the glucose, okay, so this is actually how your fructose looks like and how we convert it into Hayward projection. So now we have your glucose and fructose reacting with each other. So as you can see, it is linked with your alpha 1 and your alpha 2 carbon. So it means that the OH should be facing down in your glucose and in your fructose also because they are both coined alpha. So sa alpha 1 kung nga carbon, nag-attach ang akong glucose to its second carbon sa aton nga fructose. Okay? So, the linkage between that will become now your sucrose. Next, we have your maltose. So, your maltose comes from the combination of both glucose molecule. So, they are both connected via alpha 1 and your fourth sugar. So, in here, your maltose in here, okay, the OH is below. So alpha 1, this is the first carbon and this is the fourth carbon of your glucose on the other side. So we combine together, they will become your maltose. So alpha 1 and fourth link. It means that they are linked in your alpha carbon number 1 and in your alpha carbon number 4 on the other glucose molecule. In cellobiose, so it's not really popular, but it's the same to your maltose. It's just that it has the beta orientation for your anomeric carbon. So in here, as you can see, the OH are placed above, okay, or upward in an upward orientation. So if upward or beta siya, hindi siya maltose ang panawag but your cellobiose. Okay? So next, we have your lactose or your milk sugar. So your glucose plus galactose together linked at beta 1 and 4. It means that in my carbon number 1, my OH is placed upward and also it reacts with the other link of your on the other carbon number 4 of your galactose. Okay, so representation na para makita nyo kung paano sila nag-react together. Okay, so next, a certain enzyme is actually needed in order for us to really digest your lactose. So for people who doesn't have lactase, the enzyme that would convert your lactose into different monomers, so they are diagnosed usually with lactose intolerance. And some other countries do have this lactase mismo nga tablet in order for people to enjoy dairy foods. Okay? So number 5, we have your lactulose. So actually your lactulose is one of those laxatives that we are using in the pharmacy field. So they are the combination of your fructose plus galactose, and we have the linkage, which is your beta 1 to your fourth carbon on the next. So we have here, okay, beta in the sense that your OH is above. So beta 1 carbon and it is linked on the fourth carbon of your of your galactose. Okay, so next we have your different oligosaccharides. So the following are not really popular, but these are just the different combinations and how you would have these oligosaccharides. And for the polysaccharides, we have actually your homopolysaccharides and your heteropolysaccharides. So in a sense, your homopolysaccharide, by the word homo, it means it only contains one type of your monosaccharide or monomer. In hetero, of course, there are many kinds that would build up your sugar. Okay? So for the most popular example for your homosaccharide would be your polysaccharide, would be your glucans, or specifically your starch. So your starch is a long chain, okay, of your polysaccharide into which puro lang na glucose ang arada. Okay? So your starch do have this two form. You have your amylose and your amylopectin. So in your amylose here, we have linear straight chain, alpha 1 and 4, amylopectin. So, di na kinala na. Basta you need to know that it has amylose and your amylopectin. So, next, we have your glycogen also, into which your glycogen is a form of animal starch, or this is our storage form of carbohydrates in the body. Okay? So, it has 8 to 12 glucose units, but different ang branching. So, for your cellulose, we have also, this is also used as a structural form of your plants, so specifically on their cell walls. So, amo man iya linkage. So, it is linked via the beta 1, so carbon number 1 nga naka-beta, and on the fourth carbon on the other side. So for your dextran, we have this one, and the purpose actually of your dextran is it is a plasma volume expander. So it's one of those agent that could help if ever a certain person is undergoing shock, hypovolemic shock specifically. So it could bring up the volume of our blood so that we will not have this low BP. Okay? So it is used for commercial and pharmaceutical purposes. So dextran is commercially produced, or somehow mga bacteria takis aga produce man ni siya sang dextran. And number 5, your chitin. So this is in the exoskeleton of your crustaceans, insects, arthropods, and your some of your different pagid nga mga organism. But this is your repeating units of your N-acetylglucosamine. So we will discuss about NAG when we reach your bacteria and other pagid nga mga stuff sa inyo nga biochem microbiology. So your chitin, so I think mas makitaan mo siya sa mga crabs. Pwede mo ni siya ma-isolate sa imo nga mga crabs. And basically, ang mga shells ng aton mga crustacean, mga lobsters na bala, so they are mostly made up of calcium and units of your carbohydrates. Okay? So next, we have your heteropolysaccharides. So there are 7 of them, most popular. So sa hyaluronic acid mo, damo-damo hindi siya nag-combine. May glucose na mayroon siya mga complicated sugars together with your simple sugars. So together, they make a very complicated structure that would be used as, here, hyaluronic acid. So as you can see, this is our fluid. That is your vitreous humor, synovial fluids. So these are important fluids in our body that gives lubrications aton mata and aton mga joints. So if you look at it, they are still made up of carbohydrates. That's why it's important to really take your carbohydrates. And it's also your chondroitin sulfate. So it is found in our cartilage, tendons, and ligaments. In our skin, we have the dermatan. In our nails, we have the keratin kind of carbohydrate. And we have this heparin. Your heparin is used as a drug right now. So it is the most popular anticoagulant. Okay, so kung may mga na-stroke ka bala gulpe, kag may dako-dako kada nga mga clotting sa imong heart, you are usually injected with this kind of drugs. So it is a carbohydrate in form also. And number 6, we have your agarose. So this is produced, or this is isolated from our seaweeds via your gel electrophoresis. Okay? So I think the agarose is used also as one of the medium intergrowing bacteria sa laboratory sa aton microbiology. Okay? And lastly, we have your peptidoglycan. So your peptidoglycan is literally what we call the cell wall of your bacteria. So the cell wall of bacteria is made up of protein plus this carbohydrate, specifically the peptidoglycan. Your peptidoglycan is made up of your NAG, here, ang chitin taman bala, and your NAM, your muramic acid. So you'll learn further about that in your microbiology, but somehow these are just your overviews for the functions of your sugar. So that's it for the whole sugar mechanism. Okay, so for our lesson, we will be practicing on how to draw your sugar from Hayward projection. Okay, from from Fisher to your Hayward projection. So please study more. Okay, thank you.